Thank you very much. Bonjour. So, as you know, I'm from Barcelona, not very far away from here. It's easy by car, but by plane probably to reach Toulouse. I would like to congratulate all of you for having such a vibrant postdoc group here. This is very relevant because this is the future of science, all these young guys. And I'm going to explain a little bit cancer epigenetics, epitranscriptomics, a little bit, and examples from my lab in the last 3 years, probably in the lab. This is done in the outskirts of Barcelona, here in that white building that is the Josep Carreras Cancer Institute. I would like to start with this slide. It's something that I like a lot, the concept of identity, why we are different, why our organs are different, our cells, our tissues, why a healthy and pathological tissue is different. Identity depends on the genes, in the sequence of the genes, but sometimes it doesn't. For example, our brain and our gut, they have the same DNA sequence, but they have different phenotypes, different properties. And these are not there in the DNA sequences. What provides identity, part of that is epigenetics. These are the chemical modifications of DNA and the proteins that regulate DNA activity and the packaging, like histones. And methylation in humans, it's a very easy mark. It's only, in this case, it's only methylation of a C that precedes the guanine. This is in humans. Plants, they do other weird stuff. C. elegans doesn't have. Fly, no methylation at all, and they are very happy. But humans, we cannot live without our DNA methylation in this cytosine that precedes the guanine. Apart from this, we have histone modification that is highly complex. I'm not going to discuss today histone modifications, as I said, because it's highly complex. It will require another lecture only about histone modifications. But it's critical for differentiation, for the identity of our cells and tissues. And this is in a model of hematopoiesis, that is probably the best characterized model of differentiation. All these different steps from the hematopoietic stem cell to highly differentiated cell types, they undergo specific changes in epigenetics. And in fact, mutations in enzymes like DNA methyltransferases or DNA demethylases are able to abolish differentiation. They stop in one step of this change here. So showing that these are really relevant for differentiation. There is a special type of differentiation that happens in cancer that we call transdifferentiation. It's a way, for example, that cancer cells use to escape from therapy. This is a differentiated cell type that became another cell type without this intermediate stage of a stem cell. And for example, one way that non-small cell lung cancer escaped from the activity of tyrosine kinase inhibitors is to become a small cell that now is completely resistant to the drug. The same happened, for example, with BLL, that upon the pressure of CD19 therapies can become an AML, and now is completely resistant to the drug. This process involves epigenetics. Sorry, I think the wrong one, here. This is a model of transdifferentiation that we have in the lab. These are B cells that became macrophages under the pressure of transcription factor. And in this case, when we analyze DNA methylation, we have seen a complete change of the setting of the DNA methylome that happens in promoters and in other regions. This is an assay to measure interaction between enhancers and promoters. And you can see here that there is no interaction in the B cell between an enhancer and a promoter, and this is because the enhancer itself is methylated. When it becomes a macrophage, then this enhancer became hypomethylated, and now there is a physical interaction between the enhancer and the promoter that activates that downstream gene. So transdifferentiation, like all types of differentiation, involves important changes in DNA methylation. But in cancer, what is mostly known is that there are important tumor suppressor genes that are silenced by DNA methylation. Tumor suppressor genes can be mutated, lost by homozygous deletion or LOH, but DNA methylation and silencing by this focus methylation at promoter CpG islands is very common. And these are examples of genes in all pathways. In all pathways in cancer, there are 1-2 genes in a given tumor that is hypermethylated and silenced. But not only coding genes. Also non-coding RNAs. This is data just from my lab in the right, but many groups in the world have shown that different types of non-coding RNAs are silenced in a specific manner in cancer cells, and they contribute to the phenotype. Of course, the most famous are microRNAs. Many microRNAs with putative tumor suppressor functions, they are downregulated in human tumors due to DNA hypermethylation. But we have also others, different types of long non-coding RNAs, antisense, etc., etc. Recently, for example, we identify even that this weird, not this weird, I would say this, so until very recently boring non-coding RNA, that is tRNAs, transfer RNAs, just molecules that you just put the amino acid there, so they're really boring, they thought. They are not really that boring, and we know that they participate in the cancer process, and they are specific. And there is specific sets of tRNAs in tissues and in cancer cells, the same as ribosomal RNAs. And in this case, we identify that some tRNAs are silenced in a specific manner in cancer cells. Despite many of them bring the same amino acid, but only one is silenced in cancer cells. So this shows that they are different, they do different things, despite they bring the same amino acid to the ribosome. Okay, and this is a very, I would say, it's a very interesting finding that has to be followed up now by basic research showing what it means. This. Some of these advances has been made possible because we have tools that are very useful to analyze DNA methylation in a price-sensitive manner and pretty fast. Okay, and we don't require that deep bioinformatics. Also, this will be for the next meeting. And these are the DNA methylation microarrays that allow this analysis. And we have validated all these arrays through the years in my lab. And we have recently now, there is a new EPIC array that incorporates new CpG sites. This is the one that's going to be used for everyone. And also there is now a mouse array that is very relevant because until now, to study DNA methylation in mouse was very difficult. There was not a good system to do that. And now you can do, now we're doing hundreds of samples for DNA methylation in mouse. Important keynote for the people, when you have done this analysis, you can use the same array to determine copy number variation. So this, in the same one shot, you can look DNA methylation and gain or loss of genes because it's like a SNP array. So you can do this using the different bioinformatic pipeline. But this is relevant for many reasons. Going back a second to the cancer, it was mentioned before the hallmarks of cancer, no? And in fact, this is the last inception of the hallmarks of cancer by Doug Hanahan in Cancer Discovery, and there are many now. Every time there are more, as expected, and these are the current ones. And finally, epigenetic made it. It's there. Okay, no mutational epigenetic reprogramming by Doug Hanahan. But Doug is not completely right because epigenetics is everywhere. Epigenetics is like genetics. It's another type of regulation. So there are epigenetic dysregulation of metastasis, of cell death, of genomic instability, of senescence, etc. It's everywhere. It's not one of the hallmarks. It's everywhere, like genetics. Just to give you an example of a contribution of epigenetics in one of these hallmarks, this is an example in apoptosis. In apoptosis, there is a special type of apoptosis that now it's very fashionable that is called ferroptosis, that depends on iron. And ferroptosis, our cells, they are able to prevent ferroptosis using two pathways, one that depends of glutathione and one that depends of this protein, the ferroptosis suppressor P1 here. And this is how normal cells prevent ferroptosis. What we have discovered recently is that this enzyme here is silenced in some leukemia cells. So these leukemia cells are prone to ferroptosis because they only prevent this pathway using glutathione metabolism. And in this case, if we give drugs that target these enzymes here, these cells die very fast. So one of the weaknesses of some leukemia cells is that they are prone to ferroptosis, given these particular glutathione inhibitors. Only those with methylation of this particular gene here. Of course, I do epigenetics, and it's what I like, but I started as a geneticist like many people. And I understand perfectly well that to understand a big problem like cancer and other diseases, we cannot only study epigenetics, the tail, but we have to study the genetics, the microbiome, etc., to understand the big problem here. And we participate in many consortiums that put together all this multi-omics data. And I was remembering the one that we did with Francesco Iorio, in fact, putting together all the multi-omics we did in the cell lines at the time when he was a little bit younger. And an example, recent example of this is a study we have done with the NCI in the US. And the NCI, they had these patients that are called exceptional responders. These are people that, according to the clinics, they should die in a couple of months. But you give the regular drug, and they are alive for years. How is possible? Why they are these exceptional responders? Of course, they are a minority of percentage, but this has troubled, in some way, clinicians for a long time. So we did a multi-omics of exceptional responders. And we did, of course, exome, RNA sequencing, and we contributed to all the DNA methylation studies of these patients. At the end, the study was published kind of recently, and what our conclusion of this study is that exceptional responders, at the end of the day, they are lucky. They are lucky because they have several hits in the same pathway that is the target of the drug. For example, an advanced glioma, a glioblastoma, a GBM, that you expect to die in 1 year. So we had a patient that was alive after 6 years. How is possible that? So this patient had 2 mutations in 2 DNA repair enzymes and methylation of MGMT, that is the other DNA repair enzyme. So when you give the drug, the classical drug, Timodal, in this case, those cells cannot defend to the drug. There's no other way to escape because they have three events in the same pathway that should be repaired upon deletion by Timodal. So these patients, at the end, they're lucky. There is a trial now trying to originate mutations in the same pathway, being active, trying to create more exceptional responders. This is done in the US. If this was not complicated enough, we have embarked now in the last 5 years also, not studied only epigenetics, the modifications of DNA and proteins regulate DNA, but the chemical modifications of RNA, the other big nucleic acid that we have. And this is a very complex scenario also, because if DNA we have only 5-methylcytosine in humans, for RNA we have many modifications. We have 5-methylcytosine, like in DNA, but we have also M6A, M1A, we have pseudouridine. And this is just talking about small modifications, but we have, of course, big ones, all the circular RNAs, all the splicing, and many others. And this, here you have the modifications, and there is a complex systems of writers, readers, and erasers of all these chemical marks. This involves more than 500 genes that in our cells take care of RNA modifications. And some of them are mutated in cancer, some of them, they have monogenic disorders, etc. It's a new world. We have recently shown that in that model I told you of transdifferentiation between BALL to macrophages. So in this model of transdifferentiation, I showed you before, we have done an M6A RNA immunoprecipitation and sequencing, and we have seen a complete change in the patterns of M6A RNA in this model. So this contributes also to providing identity to the cells, not only epigenetics, but also epitranscriptomics. So now these are the genes. People are trying to find the mutations, homozygous deletion, etc. of this. And in my lab, these are our contributions in the last years. And all these are epitranscriptomic proteins that regulates different chemical modifications. For example, this one modulates RNA editing, that at the end is an RNA modification, that this is altered in non-small cell lung cancer. This regulates splicing, that it's altered also in breast cancer, etc. I'm going to give you just 3 or 4 examples of this kind of work. And I would like to start with this slide. And it goes back to the 75, okay? Great year, except for this type of pants, okay? And what happened in 1975 is that there was this paper in Nature that they show that a particular base in tRNA, the Y base, was lost in cancer. in hepatoma, was lost. This particular base in tRNA is not the base, it's the chemical modification of this here, in fact. And nothing happened until now, until 2 years ago, at the level of cancer. So everybody knows what paper in Nature, God knows what it means. Okay, 1975. But in all these years, biochemists have worked out the pathways that produce that modification. So we have 12 enzymes to take care of this particular modification in tRNAs, in a subset of tRNAs. We have 12 enzymes, so imagine it has to be relevant. We believe maybe in evolution. It has 12 enzymes to take care of that. And we have found that, in fact, what they saw here in this paper is that this gene, TWE2, is the one that is lost in cancer cells. There is DNA methylation that silences TWE2. Okay? So this is an epigenetic modification that changes an epitranscriptomic modification. And when you have this in a cell that is healthy, this enzyme here modifies this particular position, G37, in this tRNA, and this perfect match between codon and anticodon. But what happens in cancer cells is you have silencing of TWE2, and then there is a mismatch between codon and anticodon, and the RNA gets degraded at the end here. So this is a way to downstream change the expression of thousands of genes. And this happened, for example, in colon cancer. Another example that we had in the lab recently is this gene. This gene is ALK-BH3. ALK-BH3 is an M1A RNA demethylase. Demethylase M1A in the RNA. And we have seen that this is lost in cancer cells, mostly in Hodgkin lymphoma, by DNA methylation also. And what happened here is that in normal cells, happy cells, this enzyme is produced, and then is able to demethylate targets like collagens, but in lymphoma is silenced, so this chemical mark, M1A, stays there in the messenger RNA. And if you study messenger RNA in a subset of messenger RNAs, these are very stable molecules, and now they produce proteins. And that's the reason why in some lymphomas you see production of collagen, that you never expect to see collagen produced in a lymphoma cell, in a lymphocyte. Okay? But they produce these collagens because the RNA is stable, which they should be degraded in normal cells, but this is stable. And these lymphomas are those lymphomas that do poorly, because they have like a frontier that protects the lymphoma against the drugs, because you have this collagen that is very dense around the lymphoma. And you can read about this in this review in Blood Cancer Discovery. A couple of examples more. One that relates to that tumor I told you before, GBM, glioblastoma, the beast, one of the beasts in oncology, together with pancreatic cancer. And here what we have identified is that there is an RNA methyltransferase for 5-methylcytosine. So this methylates the same C that in DNA, but in RNA. And this is called enzyme 5. And this enzyme 5, when it's methylated, are those GBMs that they do well. And these are 5%. Being optimistic, this is 5% of glioblastoma that do well, good prognosis, 5%, no more. Okay? So why it goes there? What is the mechanism? We identify that this enzyme that we have is an enzyme that methylates ribosomal RNA, one position only of ribosomal RNA, and makes ribosomal RNA stable, makes a good, happy, fit ribosome there. So these GBMs, they have a ribosome that is not stable, so it doesn't produce so well proteins. We have done RiboSeq, proteomics, SciLac, RNA-seq, etc., to show that there are no changes in RNA sequencing, but there are changes in protein production. This ribosome in these GBMs is not producing proteins in the right way. So these GBM cells are almost frozen in time. They do not reproduce so fast like others. And that's the reason these are associated with good survival. The GBM is there. It's not dead at all, that GBM, but cannot divide so fast like the others. Why this is selected in GBM? Nobody knows. People think that this can be a mechanism of defense against a microenvironment that is very hostile. For example, under hypoxia, under treatment, radiation, etc. But this is associated with a good marker of prognosis in GBM. And the most recent example in this area has been done by a former PhD student, Vanessa, that also contributed to this article here, to the one in ribosomal. And in this case, we studied like a brother of enzym 5, and this brother is enzym 7, another RNA methyltransferase for 5-methylcytosine of RNA. We identified that this enzyme is lost in cancer cells by DNA methylation and silencing. And these are the spectrum. You have a lot in melanoma, but also a lot in liver. So we decided to focus in liver. This is kind of a DNA methylation sequencing. When it's methylated, it's not expressed, as you can see here, and can be reactivated. So why we focus in liver? Because liver, it's a very prevalent disease, liver cancer. It's not maybe not so fashionable like breast cancer, colon cancer, etc., but it's highly prevalent. And there are regions in the world that they have a lot of liver cancer, and they die by liver cancer. Even here in our regions, the liver cancer associated with alcohol consumption, related with the fat uptake, etc. So it's a prevalent disease. And we have shown, again, so we have an enzyme that is lost in liver cancer. What are the targets? No idea until our discoveries. So what we did is what's called RNA bisulfite sequencing. So we sequence all the RNAs after modification that distinguish between methylated Cs and unmethylated, trying to find targets in the RNA of this enzyme. And from this, we identify very few. In fact, we have an enzyme that only methylates 3 or 4 messenger RNAs. And one of these, this is RNA bisulfite sequencing. That's a very tricky technique, not DNA bisulfite sequencing, RNA bisulfite sequencing. And this is the target that is methylated by this enzyme, this particular position in this gene, a gene that nobody has heard before. It's one of these obscure genes. What is doing this gene? No idea. So you look at the gene, very few papers, 2 papers, nothing there really. So we did, I forgot to mention that the methylation there in the RNA stabilizes the molecule. Okay, makes the RNA stable. And this is shown by these experiments here. So what is doing this protein there? So nobody knows about the function of this. So we did a protein interactome, interaction of proteins to see by mass spectrometry. We found targets that are bound by this protein, trying to find some clues about what it's doing. And we found that our RNA, our protein, was binding to this one protein that, unlucky us, also very few data, okay, but the number 1, but at least there was one good data. It's a regulator of c-Myc. So at least we have a gene that has been widely studied in cancer. So here, this is the mass spectrometry data showing that. So the global idea is that enzym 7 silencing at the end leads to MYC activation, a MYC overexpression of the cells. And looking at the databases, we identified that, in fact, the methylation then predicted response to bromodomain inhibitors that usually, among them, target c-Myc. Okay? So this is the case. This is the in silico, and we put this in the wet lab. And yes, we try 3 different bromodomain inhibitors, and now those cells that they have methylation of enzym 7, they are more sensitive to 3 different bromodomain inhibitors. And this can be interesting because bromodomain inhibitors, they are great, very interesting for research, but no one has been so far really approved for clinical use because you need like a companion diagnostics, a subset of cases that are very sensitive to the drug. And I think that enzym 7 silencing can be one marker that predicts response to bromodomain inhibitors, at least for liver cancer and maybe for others. So this can be relevant because when you have methylation of this enzyme, liver cancer do poorly. Okay, so maybe this will be the patients that can respond to a bromodomain inhibitor added there in the treatment. Interestingly enough, what about the ones that are unmethylated, that they overexpress or they have expression of enzym 7? So we have seen that the ones that retain enzym 7 activity, they are enriched in this immune active subclass. So maybe these are the liver tumors that respond to immunotherapy. Enzym 7, bromodomain inhibitor, methylation here. But if you keep enzym 7, maybe they can respond to immunotherapy. So again, it can be a double interesting hit. I want to switch gears just to mention that, and this immunotherapy provides me the clue for that. So epigenetics contributes clearly to immunotherapy for many reasons. For example, we know that DNA methylation and histone modifications regulate the activity of many key immune enzymes. And we know several autoimmune disorders that they have mutations in enzymes of the epigenetics machinery, for example. So it's clear that epigenetics contributes to immune cells, etc. So can predict response to immunotherapy. Our first article in this area was done by a postdoc, one of the few physicians that came to my lab in this way from France, Michael Durousseau, that now he's a big clinician here in France. And Michael is a thoracic lung researcher. And when Michael did here in my lab, we studied lung tumors, those that receive therapy with anti-PD-1 therapy, okay? And we obtained a DNA methylation signature that predicts response, epimmune. This is a DNA methylation signature. And this is the classical study using a discovery set and a validation set here. And we found this profile that predicts response to DNA methylation, to immunotherapy. But we went further recently, and we wondered about the newest class of immunotherapy, the cellular therapy. And this is related to CAR T-cell therapy. This is patient 0. This is the first patient in the world that received a CAR T cell, Emily Whithead here. And she was here in 2012. At this point, she was very close to be dead at this point. All treatment has failed. She received the treatment, and now 22, she looks great to me, okay? And more than 10 years later. And this is a therapy with these T cells from her body that has been re-engineered to target, in this case, CD19, okay? But this therapy has problems. One, extremely expensive. So we cannot give so far CAR T cells to all patients. Second, there are some side effects, cytokine release syndrome, neurotoxicity. And third, some patients do not respond. Primary resistance or acquired resistance to the CAR T-cell therapy. So we wonder, can epigenetics predict in some way which CAR T-cells are the good ones, the one with the highest fitness? So we took, with this analysis, this academic analysis involving 3 hospitals in Catalonia, and Italy, and Israel. And these are 3 different CAR T-cells, academic CAR T-cells, against CD19, against B-cell leukemia and B-cell lymphoma. And we studied DNA methylation in the CAR T-cells themselves, just minutes before given to the patient, okay? And using that, and using this discovery validation set, we obtain a signature, the EpiCAR signature, that it's a good predictor of those CAR cells are fit or not to have a target effect, if they are effective or not. And so this can be very interesting. You can combine this with other techniques, and for example, combining with flow cytometry, we have seen that this, and also by informatic deconvolution, not only flow cytometry, by informatic deconvolution here, we have seen that this signature is in part enriched in less mature cells. It has more naive T cells and more central memory T cells. While the CAR T cells you infuse and they have very little effect, they are more mature, more committed, effector memory T cells and effector T cells. Probably this relates that these cells that are less mature, they stay longer in those patients, and they have effects in long-lasting effects, okay? So this is a way that now, for example, the clinicians, they can enrich this population, epimmune, and this is the population that should be given to the patients. These and other markers, okay? Not put all the mix of cells there. So there are particular subsets of CAR T cells that they have better effects. Just be careful also in the labs, because when we have CAR T cells, and in the clinics, if they stay longer in culture, you are changing DNA methylation. So, and finally, they can be good at the beginning, but at the end, for all the changes with the cell culture, it changes DNA methylation. This is an article that we have done in collaboration with Wolfgang Wagner from Germany, showing that there are really changes in DNA methylation if they stay for a long time in culture. So shorter time is the best in culture of these CAR T cells. These changes in culture show that these are very dynamic changes, and this can be changed by drugs, epigenetic drugs that target histone modifiers, readers, and writers. These drugs are able to target DNA methyltransferases, histone demethylases, histone methyltransferases, etc., and can be combined with immunotherapy. So you can give a hypomethylating agent that restore antigen expression, at the same time give immunotherapy that recognize that antigen there. And there are several clinical trials in this way. Not only epigenetic drugs, now that we have epitranscriptomic drugs, drugs that inhibit RNA modifier enzymes, like an inhibitor of methyl-3, that we have shown that impairs transdifferentiation. This is the one that we have. And it's not only in the lab, because this drug now it's in clinical trials. So in a very short time, an epitranscriptomic drug has made it to a clinical trial. And for DNA methylation took a long time, but now time is different. But now it's there. So we'll see if it has any effect. This is an inhibitor of an M6A RNA methyltransferase. And I want to show very little, very few, sorry, in a few minutes, some studies that we're doing with single cell, but not single cell RNA sequencing. Spatial transcriptomics and single cell for DNA sequencing and protein. And this is done because we have different technology, among them the Mission Bio that you have also somebody outside presenting this technology. The first study, this study has been led by Eduard Porta with our help here. And what we did with Eduard is wondering about the following. These hallmarks from Doug Hanahan and Weinberg, where they are in the tumor, in which location they are. And to solve that, what we have done is a spatial transcriptomics, in this case using the Visium technology, where they are. And these are different markers of the different hallmarks. And this is an assay to measure that in reality they are not random. All these different hallmarks, apoptosis, metastasis, angiogenesis, etc., they are not random distributed. They have particular sites that they are enriched in those tumors. And this is shown by this by mathematical analysis here. We have analyzed this in many human tumors among different types. So this is not tumor type specific. This happens in all tumor types. And the other thing we have seen is that location is very relevant, like buying a house. Location is critical here. And for example, there are hallmarks that are clearly enriched in the cancer cells, while other hallmarks are clearly enriched in the tumor microenvironment. For example, avoiding immune protection, immune reaction, this is more TME, while epigenetics is more cancer cells. It's more enriched in the cancer cells itself. We have also shown in this article, you can read it in bioRxiv, this one so far until it gets published there, is that this profile of expression and the space is more or less independent of copy number. So it's not the copy number that determines this location, it's the expression of the genes. And there are several data suggesting now that this goes beyond copy number. And the third piece of data I want to show you is that there is a crosstalk all the time in the space between the hallmarks. For example, if you have an epigenetic hallmark in one cell, it's very likely that the neighboring cell has this other hallmark. So there's a cross-communication all the time. And depends on the hallmark, depends on the communication you have. Another example of using spatial transcriptomics in the lab has been applied to cancer of a non-primary. This is the appearance of metastasis, and you don't know the primary site. Years ago, we developed a test based on DNA methylation because it provides identity of tissue. It's very great to provide identity, so you can give a diagnosis to the clinics. But we like it to reanalyze new samples now with a spatial transcriptomics. And these are different cap cases that we have, and the different regions. This is the stroma. These are the different clones, so I want to call it here, in the tumor of these caps here. And this is just a clinical case, for example. This is a patient that had a metastasis in the bladder. But the medical doctor didn't know the origin. What is the primary site here? So we took this case, and with the spatial transcriptomics, this is the stroma, and these are the different regions that we have here. And we can compare these patterns with the known patterns of RNAs transcription that we have available in the literature. And now this is type of comparison here using the Visium technology. And we have identified in reality that bladder came from a pancreatic tumor. And that's the reason the medical doctor did not see it, because usually pancreas do not give a metastasis in the bladder, give metastasis in other places. But now the molecular biology, this type of analysis, is telling you where it came from. It's an application of spatial transcriptomics. Another application of this type of technology in another article, in this case, is what we use MDS. MDS, it's like a leukemia type. And we have, it's treated with an inhibitor of DNA methylation and hypomethylating agent. And we have taken a sample at the diagnosis and after finishing the treatment with the inhibitor of DNA methylation. We wanted to see the changes at the single cell level of the clones. And if this predicted the response to the drug or not. And we have done single cell sequencing of DNA, of target sequencing in this case. You can sequence around 80 genes, you can imagine, okay? But at the same time, in the same cell, single cell protein sequencing of around 40 proteins. You cannot do all. It's 80 genes, 40 proteins at the single cell level. And these are some of the analyses. So there are MDS that upon treatment, they have this evolution, and some they have this evolution. Yes, I have 5 minutes, no? Yeah. So, and in this case, they show tumors that usually, MDS that respond very well to the therapy, and tumors that don't respond to the therapy. If you do the fish plots here, you see that these tumors here on front, they respond very well. The mutant clones get reduced, almost nothing. But those do not respond, they have this weird stuff, or they do not care. The mutant clones do not care about the drug, or there is an expansion of mutant clones. So the drug benefits the tumor sometimes. This is, you go at the protein level. This is the UMAP of all the proteins, and you can select populations that they are enriched in these particular responders or non-responders using this type of approach. But of course, you want to combine protein and mutation. And this combining both, for example, you can analyze which cells particularly carry the mutations that we describe. And this slide summarizes a little bit. And these are the populations that the mutant cells are the ones that predict response. This patient, for example, has all these mutations at different cells here. And these are the cells. It is in CMPs. And you can see that if respond, they disappear. In the resistant, they stay there. So you can try to predict using this technology. And just to mention that you can do single cell, of course, of DNA methylation now. That is very tricky, but you can do it. We do it in the Dan Landau style, that he pioneered that. And this is single cell DNA methylation at the moment of diagnosis of MDS. Upon treatment with 5-azacitidine, this is an expansion of single cell mutation, methylation heterogeneity. And sometimes these patients, they develop AML, leukemia. And now it's a point again. Very few clones with methylation that is very homogeneous through. So you can do single cell with many things. And now I would like to finish with 1 minute, sorry. And just for human curiosity, I think that this is an article that also has received a lot of attention. Identity. What about, we showed years ago that twins, they have the same DNA, they show epigenetic difference. And this can explain discordant phenotypes in twins for diseases. Okay, this was shown many years ago, and now it's fully accepted that. But what about that people, they look alike, that they are similar, but they are not related? So we have done, this is a project that took forever, took two postdocs or 3 postdocs to do, okay? So we look for people in the world that they are extremely similar, but they don't have any relation between them. They even do not know each other, okay? So, and we did a multi-omics analysis of these people. So we analyzed, so first we did a facial recognition algorithms, the same that used by police and the airports, to show that really in a mathematical way, they are look-alike, okay? Sometimes they can open the iPhone of the other. Just, they're very scary, okay? In this case. And we did a multi-omics. In the multi-omics, it has DNA methylation, DNA sequencing, and also has the microbiome and copy number. And we saw that in this case, what's genetics? Genetic composition was similar between them. The SNPs, they share a higher number of SNPs than expected, a higher number. So they look alike because they share SNPs, more than expected in normal population. Of course, in this case, the SNPs relates to the face morphology, okay, the structure of the bones, etc. So, and this is, I think this is pretty interesting. For 2 reasons, and now it's 10 seconds. One, for the future, is that now for the first time, we have tools that from genomics, from the sequence, you can start making faces, okay? Drawing faces. So this is an interest in forensic medicine. And we have already received calls from police agencies trying to help in cases. Okay? Of course, it's very difficult. From a genetic sequence, I have to draw a face from this. So it's not easy. But it's possible to do it. It requires a lot of bioinformatics, of course, and artificial intelligence. But now, for the first time, you can do these kind of things. Not only that, but you can do the other process. Now, from the face, you can deduce or infer genetic sequence. Okay? So you can induce, do some screening of diseases, even in childs, even in little kids. So this is the other process around. So I think this is work for many generations and many informaticians working on that, because, but it is a great material to work. I would like to finish here and thank all the members of my lab and our supporters. Thank you very much.